Okay. Yeah. Hello everyone. Today I'm talking a little bit about sRNA, like single cell RNA sequencing. And I'm trying to like make it end to end, where I'm like going through like the beginning, like to how it starts to completely all the way to the end, like the analysis you do on it, and also like trying to um, yeah, talk about it in like a very pragmatic way that like you can actually use it as well, and like apply this the knowledge and whatever yourself, and like also try it out yourself, like the analysis, etc. Yeah, so first of all, why? Because I mean, very simple, because it gives you like single cell resolution. Of course, I mean, this is kind of basically the definition. Um, but the interesting part, I think, is like that it like it partially only, of course, shows you the inside of the cell. So it kind of, if you think of it in terms of like engineering principles, it kind of helps you to like break down, um, like yeah, in engineering, like you would, for example, try to break down a radio and then like try to rebuild it from those like bottom parts, and this is how you would also like learn how to understand it, and then you can also improve upon it. And this, it kind of goes further, like helps us to like um, yeah, enables us to do this a little bit further, because you can, like, I mean, then you also like in some ways what is missing, of course, there is like the spatial part, which people have been like starting also as well, but like, yeah, so basically that you can kind of have the tissue, you can look at the tissue, how it's like structured, like the all of the collagen, the proteins, the glycoproteins, whatever, outside, and then you can also have the cells, and you see them outside, and then you can also see inside them. Yeah, and so what is needed for this? Okay. Okay. Yeah, what's needed for this? Basically, you have like the chromium 10x chromium series barcoding machine, and then you also have like you need a sequencer, um, and of course, like you don't need the chromium X. It's just one thing you can use, but like if you will look at the research, it's by far the most popular thing. So I'm just going to focus on this. Of course, be aware there are other solutions for this also, for the barcoding. And yeah, the sequencer, other sequencers exist, of course, as well. Um, this is just also a very popular way for the analysis. And yeah, so of course, and then you will need many other things as well, but I will just assume like either you have those, or this is kind of rather inexpensive to get. I mean, for example, you, for each assay you will need like yeah, you will need the suspensions, and you will get them as a kit from 10x as well. And yeah, I mean, then the equipment also, but yeah, I assume, like, I mean, for example, for the analysis you will need some software as well, but I will go into them, yeah, later. And also all of them, like most of them, many of them, like almost all of them are free. All right, and then yeah, so imagine you have now the idea, you want to like have, you have a tissue, and then you're deciding, I want to analyze this. Um, then of course you have like all of the microscopy, and then if you go to single cell RNA sequencing, you, I mean, either you have like a tissue, then you kind of need to prepare this. There's um, no general way of like breaking down tissue and then using, like getting a single like a cell suspension, um, but and this is, yeah, yeah, so this is what 10x also writes. but yeah, so once you have this, then like you need to kind of find a protocol for this, and will break it down, um, and and then you will like, clean it up, and then you will kind of like do some quality control before you also um, before you do it, like before you try to like sequence it or add the barcoding. And then if you have the cell suspension, I mean basically, I mean this kind of goes over into this as well. Either you have them frozen and you have to thaw them, if yeah, and then once you have like you either have like the general sample preparation, and in this case, like if so basically if you have enough cells, and then how you do it basically, it's just like changing the buffer and making sure that the concentration is correct. Cell counting will then just ensure that you have like over like 300,000 cells. Yes, and so this is kind of the step, and then you just put it like you add the cells, the suspensions, and everything. That's like a kit. You can watch a YouTube video for this as well, and I will also link this. And then you will insert this into the barcoding machine, and then what happens inside there is basically this thing, and yeah, so you add this like here those gel beads which have those things attached to them, and this it's sort of like a it's like a how's it called? Like it has like lysis things as well. Yeah, so like it has everything to yeah, well, okay, one second. Yes, I have the gel beads, and then they are like kind of flowing in this direction, and then you also add the cells and enzymes here, and then they will get together, and because here you will add the oil, they will kind of group together into like one cluster, and then the nice thing about this is here inside this like this sort of cluster bubble, you will have you will have all of the required things for it to like kind of do the reverse transcription and lysis as well. So all of the cells which are in here, they will get lysed, and then the RNA will be released, and this RNA attaches then to this bead, and that will be reversely transcribed to cDNA, and then in the end you will have this, and like a lot of like a 10x barcode, and then the UMI, which is required to assign the read to a cell or molecule, whatever you're using. Yes. And so then you have all of this, and basically you get some outcome data. Either you will use this directly, and then later, if you publish your study, you will probably upload this to Geo, and you upload this to Geo. If you're interested in like doing analysis on something which is online, you will have to use, in most of the cases, you will have to use Geo to download it. In this case, you will like download the file here. Like okay, so basically, let's go briefly through the structure. I mean, this is kind of structured in. You have like everything is in the GSE. Like in the GSE, it's kind of the like the hubs sort of thing. Like this is the kind of the ancestor, or not the ancestor, but like this contain like on this is basically all the other information. So it's kind of like an entry node, and this GSE stands for Geo Series, which is kind of like, I mean, it can be multiple things. For example, this is now just like one publication, and then you have different different platforms and samples. Platforms, mostly right now it's just like the sequencing machine, uh, and barcoding, I think. And when you click on this, you will see that you will see like all of the experiments which have been, all of the samples which series which have been which use this platform. So this is not specific to the series, like the number is the same for different series. And then samples, a lot of times you will have multiple samples. Here you just have one, and they will like the different tissue or samples for, yeah. I mean, I think this is self-explanatory. And then also relations, and this is kind of interesting, the bio project, and this is kind of like the even more top level summary of the study or the series, because here it's just the like the not the sequence, but like the assembled and like finished data, like processed data is on uploaded here, and here on this you will find like all data. Yes, and then you can download this and unzip it, and what else can you do with it? Yeah, and then you can like go over to analysis. If you use 10x, you will probably use the cell ranger software, and the cell ranger software outputs like different files, like the barcodes and I need to check, like a barcode, the read count metrics, and then also one second. Barcode, yes, and the features. So there's free, and from those you can very simply transform this to an H5AD file. H5 files are like kind of just, I mean, I think people are familiar with this, but like H5 files are the files which are like organized basically in sort of like a hierarchical way, and it's and then the AD is kind of just a specific type of structure of those like hierarchical of this hierarchical thing. So you have like specific categories which have to be in there, and they have to be in there because then they can be like used down in further like downstream analysis. I mean, because the tools which you use, for example ScanPy and Surat, they require like a specific type of format. And then you can convert those three files to an H5AD, and then you can use them for further analysis. I just realized my image has kind of, here you go. I think my image was too big. I'll also move it to the right. Yes, so those are the things you might have not seen. This you have to download. Alright, and then the use cases of it, we know why we do it. Now, how can we use it? 1 second. So the use cases, cell type identification. This is kind of so okay. Cell type identification. You can, and there's a very good tutorial by a person which I will link. Like you can do like dimensionality reduction, and you have like by PCA or like UMAP, and then you can, for each cluster, you can figure out like marker genes. So like genes which kind of mark the specific cluster, which are unique for it. For example, because it's very highly expressed in this cluster. And then you can use those genes to identify the cell type. And you can then, for example, also create like antibodies, antigens for it. I always confuse those two. Yeah, so it's antibody. So anti-gene is the protein of interest, or like you will have this will have like the epitope to which your anti-gene will bind. Anti-gene was correct. No, sorry. You made it right. Yeah, the protein of interest is acting as the anti-gene, and the antibody binds to the anti-gene. Alright, now we have it. Good. Which you can then use to like also recognize like the population in vivo. Of course, like this has like multiple use cases. You can use like the marker genes also for like cancerous cells or carcinogenic cells. Yes. And cell hierarchy reconstruction, like if you do it in like in a specific time, like with a time series, you can figure out what leads to fate A and what leads to fate B, and then you can also infer regulatory networks. For example, if there's like a correlation or something between two to the expression of two genes, which you of course cannot do with bulk RNA sequencing. Yes. Analysis for this also, once you've done this, once you, for example, also found out marker genes or like you did the clustering, nice way to visualize this is also, visualize this is to upload it to the celix gene, celix gene, I think it's called analysis, but it might be called something else. Celix gene. Explorer, I think. Yeah, because it's not necessary, but like it has nice functionalities. For example, in circle this, figure out marker genes like this, I think, if I remember correctly. Also, you can color it nicely. If you, for example, have this, you can kind of visualize it a little bit better. And yeah, I have to be honest, I haven't like figured out the full utility, how to use it, like maximize the use of this. But yeah, this is kind of the basic. Those are kind of the basics for it. All right. Yeah. So quickly, I also made like a summary of like scRNA and Surat. You can read through it. Um, it's just that Surat and scRNA are kind of competing softwares, and scRNA is better for some things, and like for big, yeah, for big data. Like if you have a lot of cells, and then also it's connected well to like ML libraries. It's usually used there as well. And Zerat is more if you like. Also, this is in Python, as the name suggests, and Zerat is with R. Yeah. So I mean, you can use either, um, but yeah, I mean, especially now, I think with large language models, it's not too important which you're using, because yeah, you can like, I mean, even if you don't know R, you can like ask ChatGPT how it works, and then yeah, you have this. All right, and this is kind of it. I hope this was helpful. This should just serve as like a brief, like I said, end-to-end introduction, and I will try, like I will probably make some tutorial on how to like do it specifically. There's a lot of papers right now which I'm reading, or like YouTube series, lecture series which I'm watching, and like I will try to like compile something out of this as well, because yeah, in my opinion, this was a little bit too top level, this video, and I would like to like give everyone also a more in-depth video, and like specifically, for example, we kind of I kind of brushed over how to use it, um, but I would really like to make it more specific, that you can actually know the caveats, for example, of using one of those things, like of of like identifying cell types, and like how you would do it in practice, and then how this would then be useful for like the next iteration of things you're building. Specifically, my interest lies in the tissue engineering, so it would be interesting for me to see um, how how you can like use this for example for benchmarking the engineered tissues, because I think single cell RNA sequencing